Hello, I'm Matt Schlesinger. I'm the Chief Zoologist for the New York Natural Heritage Program.
And I'm Erin White. I'm a zoologist and project coordinator with New York Natural Heritage.
And we are thrilled to have the opportunity to talk to you about the Empire State Native Pollinator Survey, which is a project that we have co-coordinated over the last 5 years. Our study began in 2016 based on the New York State Pollinator Protection Plan. This is a fortunate protection plan that came with funding that went to the state, and the state passed that funding to our program to design a multiple year pollinator survey to evaluate the status of native pollinators. So we designed that study from 2016 to 2017 and secured funding for the study itself, did the primary field work in 2018 through 2020, and then we are now in the process of finalizing specimen ID and integrating a whole lot of data sources, ranking species and analysis and writing. So in fall 2016 through winter 2017, we designed the study, and I include this because I think it was an important part of the project, is to spend adequate time figuring out what we wanted to do. First thing we wanted to do was find experts. We were not expert in many of the taxa that we wanted to include in the project. And so we assembled an all-star advisory committee, including some familiar names from academia and federal and state government, also nonprofits and museums. They helped us through a workshop and a series of conference calls, decide on a project goal, what our target taxa would be, the sampling design, and field methodologies. And then we wrote a study plan. We wrote that study plan that in 2017, and that influenced the budget that we submitted to the state and got funding for the rest of the study. We came up with a goal statement together to determine the conservation status of a wide array of native pollinators in non-agricultural habitats. Non-agricultural is in there to separate us from the pollinator funding that went to honeybees. And so we focused on native species in non-agricultural habitats. And by conservation status, we mean in natural heritage and nature serve lingo, the S rank. Many of you are probably familiar with S ranks. And we rank species from S1 through S5 in New York State. And the three components of that are rarity, trends, and threats. So if you work backwards or from the bottom up in this diagram, you'll see that information on distribution and abundance influences rarity. Comparing that with historical distribution information gives us some feeling of trends, and then scope and severity of threats, or a measure of intrinsic vulnerability, is the threat component of conservation status. And then we had a variety of methods that we used to come up with each of those status factors. The first really important part was to figure out which pollinators we wanted to survey. The state said, do a pollinator study, and we had to narrow it down because there were thousands and thousands of species. So I'm going to turn it over to Erin now to talk about that, and then talk about a bit more of our study methodology.
Okay, thanks, Matt. Yeah, so this was one of the first things that the advisors helped us do, was really deciding on which focal species to focus on for the project, and to highlight the importance of a variety of native insects and their role in pollination. We really wanted to focus on those primary insect orders that are known to pollinate native plants. So that includes Hymenoptera, Diptera, Lepidoptera, and Coleoptera. So within these orders, we selected species or groups of species that have been documented or suspected to be important for pollination. We also selected species that have poorly understood conservation status, and we wanted to make sure our list contained known or suspected at-risk species. We also wanted to make sure that we selected groups that are taxonomically stable and weren't so diverse that the goal of reaching conservation status for each of the species within it wasn't unreachable. And we also wanted to make sure that we selected groups that could be readily identified to species level, since that's the level that we needed our data to be at in order to get the distribution of the species across the state for that rarity information that Matt talked about. And we also wanted to make sure we included some groups that could appeal to our community scientists or citizen scientists, such as those that have charisma, or also those that have field guides written on them. Next slide, please. So for our Hymenoptera, we ended up selecting Bombus and Melisodes within the family Apidae. We also selected Megachile and Osmia within the family Megachilidae. We also selected oil bees in the genera Macropus and Melita. And we have 5 species of very rare bees in New York in these groups. And then a larger group of mining bees in the family Andrenidae, Andrena and Calliopeus, of which there are many species that are believed to be in decline. Next slide, please. For our focal flies, we focused on 2 groups, one of which were the Serpidae, the hoverflies. This is a really large group in New York, over 300 species. So we wanted to sort of narrow that down to a group of about 115 species that are known to be saproxylic. This just means that they spend part of their life dependent on dead wood. And these are known indicators of late successional forests and are important pollinators of spring ephemeral plants in those forests. We also focused on a group of bee flies in the family Bombyliidae, genus Bombylius. One of these species is pretty common in New York, but the other 9 are very rare. So these were our 2 focal fly groups. Next slide, please. For our focal beetles, we selected 2 groups, one of which was the flower longhorn beetles in the family Cerambycidae, subfamily Lepturinae. And there are about 100 species in this group. And then a much smaller group of hairy flower scarab beetles, 5 species in the genus Trichiotinus. Next slide. For our focal Lepidoptera, we focused on 2 groups of moths, the Sphingidae family, which are the sphinx or hawk moths, and also the flower moths in the genus Shinia in the very large family of Noctuidae. And there are some diurnal species in this group, but most of these are nocturnal species. You'll see in a sec that most of our survey effort focused on diurnal surveys. So we really relied heavily on our community scientists to collect photographs of the nocturnal moth focal taxa. Next slide. So in order to survey for these different focal taxa, we came up with 3 complementary survey approaches. We conducted surveys. The heritage biologists conducted surveys using 2 different sampling techniques of bowl or pan trapping, as well as hand netting. We also had a community science aspect of the project where we trained volunteers at weekend workshops in all of our methodology. And many of our volunteers ended up submitting photos as vouchers for their observations, although a small enthusiastic group was trained in a bee course given by the Danforth lab at Cornell, where they gained the confidence to collect specimens. But all of the efforts from the community scientists really helped us fill in the gaps of and get to more places in New York that we wouldn't be able to otherwise. In addition, our heritage biologists visited several museums that housed large insect collections from New York. So not necessarily museums just in New York, but also other places as well, where they had New York records on our focal taxa. And this really helps us get at those historical locational information for the focal taxa and that trend information that Matt was talking about earlier. Next slide, please. Okay, and this was just one type of survey that our field biologists completed over the course of the survey. Our extensive surveys were basically a set of points that were randomly distributed on protected lands, stratified by ecoregion, to cover the diversity of habitats across New York State. We were able to get to 150 different sites as part of this framework over the course of the survey. Next slide, please. At each site, so this would be a day of sampling at each of these extensive survey sites, we conducted a sample at each of 4 habitat types. And we tried to include these 3 habitats at each of our sites: forest, meadow, and wetland. And we also tried to include a roadside if it was available. We selected habitats that were within 250-meter radius of each other so that they were accessible within a day, within walking distance. And at each of the transects, we set up a 42-meter line of bee bowls. So we had 15 bee bowls laid out in that line of fluorescent blue, yellow, white, and would leave them out for 5 hours during the heat of the day. And then throughout the course of the day, our 2 surveyors would survey for 30 minutes, doing targeted hand netting at flowers for our focal taxa. In some cases, especially like the Adirondack ecoregion, we didn't have all of these habitats available to us at every site, but we may have had 2 habitats, so we could supplement the sampling and do 2 forest and 2 wetland stations at the site. So we tried to have the same amount of effort and have those 4 stations at each site. Next slide, please. To really supplement our extensive survey effort, our advisors recommended that we also do a little bit more effort for those focal taxa by looking in specific habitat types that we believed, or our advisory committee believed, could harbor these rarities. So those included alpine, peatland, late successional forest, coastal dunes, and barren habitats. And we relied heavily on some of our partners to help us get to some of these places like alpine and dune and barren habitats. But we did the same sort of protocol, bowl sampling and hand netting at each of these habitats, only we did 4 transects within the same habitat type. We tried to get to 10 to 15 sites each year. With the late successional sampling, we also employed another survey technique called malaise sampling because we were targeting syrphid flies in that habitat. Our heritage biologists also did target species sampling, and this would often entail going to historical locations for our focal taxa to try to document if they were currently still there. And we also did a number of de novo searches in habitat types that we believed could harbor some of our focal taxa as well. Next slide, please. And then, as part of this project in the beginning, we really wanted to kind of have a one-stop shop guide for all of our participants to use on the project. So we created a participant handbook that just contains all of our methodology, all of our protocol, data sheets, how to submit data, and also how to process specimens, how to upload photographic vouchers. We have an example data sheet here on the right that contains all of the information that we wanted to have collected when people were collecting specimens, so that we could tie all that information to a given specimen and make it into a record and be able to do our conservation status assessment. We utilized iNaturalist, which I'm sure many of you have heard of. It's a great online platform. We had a project within iNaturalist for the Empire State Native Pollinator Survey, where people could submit all of this same information. But if they were just collecting photos at a site, they wouldn't need to fill out a data sheet. They could just submit all of that information online on iNaturalist. And back to you, Matt.
Thanks, Erin. If you're admiring our project logo, it was designed and drawn by Sam Verocchi's daughter. So thank you, Anna. I just want to touch on key partnerships we've had during this project because they are critical to the success of the work. And so I'll try to do this quickly, but the New York State DEC provided funding and also advice along the way. And SUNY ESF was necessary for hiring. We're a SUNY ESF program ourselves, and we hired our field crews through SUNY ESF. And members of both DEC and ESF were on our advisory committee as well. At Cornell, of course, Brian Danforth's lab has been doing almost all of our BID and leading workshops. They've been a huge help and great partners to work with. The US EPA has funded a study on wetland pollinators as part of a wetland program development grant. SUNY Cobleskill has the Greenwood Lab, who has taken on the majority of the fly identification and sampling. And then the Central Pine Barrens Commission from Long Island has sampled the heck out of the Pine Barrens the last few years. And so we'll have a great data set from that really important region for pollinator diversity. The Adirondack Mountain Club has summit stewards that protect the rare plants from trampling on the high peaks of the Adirondacks. And so they volunteered to help us sample up there, which was a huge help for getting to these remote places. And then New York State Parks allowed us to sample in some of the pristine habitats that the New York Park System manages as part of our existing MOU with them. So leveraging those two projects off one another. And I touch on these because they were so important for the success of our project that I didn't want to leave them out. And we had a really successful project, we think. We have 35,000 specimens and 35,000, I don't know why it's the same number, iNaturalist observations as of last count. And all over New York State, you can see the iNat observations underneath everything in kind of pale tan pink, and then the sampling for specimens in the extensive surveys and other collections and target habitats are on top of that. And ID is ongoing with a target of finishing up by September of this year. I wanted to address some things that maybe we would have done differently. This was a special request from Hollis on what we would have done differently, and I'll have to say that we don't really know yet, and we won't know entirely until we've analyzed the data and determined just how many species we can do conservation status assessments for with the data we've collected. So ask us again in a year, we might have a different answer for you. At the moment, we're very happy with the amount of sampling we were able to accomplish, even with the global pandemic, and the amount of volunteer and citizen science effort we were able to cajole out of people. It's been a really special opportunity and project for us. There were some things in our wish list and our study plan too that we had hoped to be able to do. Those included nocturnal surveys, looking for especially moth pollinators of plants in meadows and other open habitats. I think critical for a monitoring effort would be some better estimates of abundance than we were able to get. We'll be able to get some index of abundance from the bull trapping, since we standardized the amount of time they were out and kept track of start and end times for bull sampling. But our hand netting, as Erin mentioned, was targeted, on flowers, so not a sweep net that would be repeatable for getting abundance estimates. But I think for a monitoring effort, we would like to get abundance estimates if possible. We also didn't collect any habitat data, and that was a difficult discussion with our advisory committee, and difficult for me as a biologist to realize we were just going for species records, the occurrence of a species in a place at a time. That was the primary piece of data we were looking for, and we prioritized that information above all else, including host plant use or other habitat associations, which felt like secondary. And so we knew that any time we spent collecting data on habitat was time away from getting species occurrence information. For a monitoring effort, though, I could see very much that you would want to know if a fauna is changing over time. You want to be able to determine if there are changes in habitat might have driven those. Fourth, the community science effort we employed was pretty freeform, and we allowed people to go wherever they wanted, and we're just very happy to get the involvement that we did, but it wasn't structured in any way, like a breeding bird atlas or some other effort might have been, or the breeding bird survey. This was very much, go where you will. Here's some suggestions. Here are the focal taxa we're interested in, but we didn't structure it in a way to spread the effort out across the state or spread the timing out. So that might be something to do differently if for a monitoring effort, if there's a citizen science or community science component. And finally, just a plug for the possibility of change in status, like say from S1 to S2 or from S3 to S1 as a monitoring metric, because abundance can be so hard to track in a highly variable insect populations that potentially these broader changes in status might be a useful metric. And that's all the slides we have. Thank you very much for your time and the opportunity to present to you. We have a website where you can learn more about the project if you like, and we look forward to participating. Thank you.
Thank you.